The overall goal of this procedure is to obtain enriched mouse embryonic motoneuron culture. The procedure begins with first sacrificing a pregnant mouse, removing the embryos, and dissecting the lumbar spinal cord. The next step is to dissociate the spinal cord into a single-cell suspension by enzymatic treatment and trituration. Following this, perform the lectin-based purification for separating the motoneurons from the cellular remainders. Next, after estimating the total cell number, the cells are plated on pre-coated dishes or cover slips. The final step is to adjust the density of the culture depending on the experimental procedure. Ultimately, as the cultured cells grow, typical motoneuron morphology can be observed.
For the success in preparing and culturing mouse embryonic motoneurons, it is essential to test different materials, substances and solutions, as the cells may react sensitively to any of these in changes.
The motoneurons can be cultured in high density for the isolation of RNA or proteins. However, there is a possibility that the cells will build up active neuronal nets which lead to the survival independent of trophic support. Therefore, in some cases, it might be better to culture the cells in low density for RNA or protein analysis. Demonstrating this procedure will be Rebecca Conrad and Teresa Sczepan. Both are graduate students in our laboratory.
Begin this procedure by preparing the working solution of 0.5 mg per milliliter polyDL-ornithine hydrobromide in 150 millimolar borate buffer with pH 8.35 and sterilize the solution by filtration. Next, sterilize the glass cover slips by flaming them in 100% ethanol.